In this second video for genetic engineering and biotechnology, we're looking at IB 4.4. We're going to take a look at some of the other topics of genetic engineering and biotechnology. Again, some really interesting topics and applications of some of the things that we looked at in the previous video with gel electrophoresis, PCR. In this video, we'll take a little bit closer look at cloning and how we can really modify organisms, genetically modified organisms. We'll take a look at those as well. So to start out, we want to get a definition of what a clone is, what makes something a clone. And a clone is a group of genetically identical organisms, or a group of cells derived from a single parent cell. And if you think about that definition, that really can apply to identical twins, because an identical twin, identical twins start from a single fertilized egg cell, and that egg cell splits into two separate cells. And so traditionally an egg cell is fertilized, and that cell goes on to divide and to grow and to eventually produce an offspring, a fetus. With a genetically identical, a twin or a clone, that cell splits, and then each of those two go on to divide and to grow into two identical organisms. And so here are obviously some pretty famous twins. The first two being Dolly the sheep that was cloned, which we'll talk about here in a second. Genetically modified organisms, slightly different than a clone here. They're organisms whose genetic material has been altered by some sort of genetic engineering technique. Glowfish, which we talked about earlier in the year, are a great example of genetically modified organisms. There's genes that can be turned on or off to make organisms glow. These genes are oftentimes derived from marine organisms like jellyfish or some type of anemones naturally glow. And if that gene from those jellyfish or from those sea anemones can be extracted and put into other organisms, like some of these fish, typically zebrafish have been used to make glowfish. If that glowing gene can be put into the zebrafish, it can then glow, like we see in commercially available glowfish. Other organisms that are often modified are plants, crops. Corn is one of the most genetically modified organisms on the planet. We'll look at some of the advantages and disadvantages of that. And in looking at genetically modified organisms, the reason that it's possible to modify these fish or the corn, and looking at this fish for example, the sequence of DNA in those fish consists of adenine, thymine, guanine, or cytosine. Now, the gene that is inserted either from sea anemones or from jellyfish is made of the same material. It's also made of a sequence of adenine, thymines, guanines, and cytosines. And so because of that, all organisms on the planet are sharing the same genetic information. It's arranged in slightly different order and contains different amounts, but because it's the same, organisms can transfer, genes from organisms can be transferred from one to another because that genetic code, those adenine, thymine, guanine, and cytosine, is universal. It's the same in all of the different organisms. So here we've got some bacteria, some zebra, looks like a little garter snake, a really beautiful clam. All of these organisms are comprised of the same genetic code. Now, obviously, the order of that genetic code varies quite a bit, and there's different amounts. If you were to throw in humans into this group of organisms here, ours would be quite a bit different than most of these, probably a little bit more similar to the zebras than, say, the gardener snake or the clam. But because we all have the same basis for what makes up each of these different organisms, because we all share these adenine, thymine, guanine, and cytosine, it's possible to transfer or translate these genes from one organism to another. And so let's look at some uses of genetically modified organisms. One example would be inducing salt tolerance in tomato plants, actually modifying tomato plants so that they can grow in an environment that is in higher salinity amounts. Another example would be producing rice that have beta carotene in order to be able to produce vitamin A. Another example would be crops being able to resist different bugs, basically having built-in herbicide resistance inside of the crops. And another example would be producing milk, sheep milk, for example, that has factor 9. That's a human blood clotting factor. So sheep would produce milk that have this factor already in them in order for patients, humans primarily, to be able to have this factor 9 in their bodies. And so there's some definite advantages to genetically modified organisms, but there's some definite potential cons as well, some problems and some concerns with genetically modified organisms. And we're going to take a look at these in more depth in class and have a chance to really discuss these, because there's some really positive advantages and some really negative advantages as well. So some of the potential positives. One, increased crop efficiency. You think worldwide, hunger is probably one of the biggest issues affecting the global population. Yes, we have a huge number of humans living on the planet, over 7 billion. How can we continue to feed this population? Even in the United States, we have thousands, hundreds of thousands of people who are going hungry or not getting enough food. There's only so much land on the planet. How can we improve or solve this problem? And really, this is looking at the efficiency of how much food is able to be produced per land. By modifying organisms, we can increase the overall production of crops, the overall efficiency in producing crops. As I said earlier, herbicide tolerance, improving the quality of crops, production of biofuels. Fuel is a major topic, especially in the United States. Use of gasoline, looking towards cleaner sources of energy, biofuels being a potential source of energy that could be used for machines and for vehicles. With all of these pros, and I'm sure that if you were to do a bit of research, you could probably find quite a few more. There are also some concerns, and one of those, and a big one, is the inadvertent transfer of allergens, unforeseen effects on non-target organisms, and the escaping of modified genes. And that's the one that I want to focus on. Genetically modified organisms have had their genes modified. Literally, the genes in those organisms have been modified or changed, either by inducing or introducing genes from another organisms, like the glowfish, or by modifying the sequence of a specific gene. In one way or another, these organisms have had their genes modified. And in looking at corn or crops, for example, if these different crops are genetically modified, what is to stop the genes that are modified in specific crops transferring to other crops? There's nothing to stop that, and that is And so the next thing that we want to look at is how we can actually clone organisms and how this process actually works. And as you probably know, one of the first successful process of this was in Dolly, sheep that was cloned. And this process is called nuclear transplantation. We're transplanting a nucleus of one organism. And when you think about cloning, if you think about sci-fi movies or, usually on television, comics, whatnot, you think of somebody walking into this machine and out comes two identical individuals that have the same personality and the exact same looks, etc. Well, that's in the movies. In real life, it's quite a bit different. And how this process works in real life is, if we have a individual that we take a nucleus from one of their cells, and we have a second individual that we remove a cell from, and from an egg cell, from a female. And if the nucleus in that egg cell is removed, we can combine the nucleus of our organism that we want to clone with the egg cell that we've removed from the second organism, the donor. By combining these two, we have an egg cell that has the genetic information, the nucleus from the sheep that's going to be cloned. And then by taking that fused cell, begins to divide, we get an embryo forming. And by taking that embryo and putting it into a foster mother, eventually that embryo will continue to divide and develop into a new individual. This is our cloned lamb, Dolly. And this is the process of nuclear transplantation. This is how Dolly was cloned, and one way that we can clone organisms. And we'll look at this a little bit more closely in class and discuss this application a little bit further. Another process that we want to look at is transferring genes, specifically by plasmids. This is one way that we can transfer genes from one organism to another. So kind of in thinking of the idea of how genes could be transferred from a glowing jellyfish to a zebrafish to make it glow. And again, keep in mind that this is possible, as we discussed, because all organisms share the guanine, cytosine, adenine, thymine nucleotides. And so by using E. coli, we can transfer genes from one organism to another. E. coli contain plasmids. They're bacteria, and so rather than having an X-shaped arrangement of their DNA molecules, they have a small circular arrangement of their DNA. And we call those plasmids. The plasmids can be removed, and they can be cleaved or cut by specific restriction enzymes that have been identified to target a specific area. So in that plasmid, that circular DNA, we can target a specific area that we want to identify and to cut. And what I mean by cutting is we're actually going to cut out a portion of that plasmid and replace it with DNA from another organism using the same restriction enzyme. So we've got a plasmid, we're going to remove a portion of that plasmid, of that DNA, and insert that same region of DNA, that same gene from another organism. So it's put into that E. coli. The DNA fragments added to the open plasmid are spliced by DNA ligase, and then we can insert that recombinant plasmid inserted into new host cells and cloned. And so let's take a closer look at this in a nice image here. Here's our E. coli bacterium. Here's the plasmid, the cleavage site, the portion that's going to be cut right here. And so that portion is removed. Inserted is some foreign DNA of a specific gene that we want to look at, is inserted into the plasmid. That plasmid is reinserted back into the bacteria. The bacteria is used to insert that foreign DNA into the chromosome of a plant cell, for example, how we could actually modify plants. Plant cells are grown in culture, so they're actually grown in the culture, and then as they begin to develop, they eventually develop into a plant, and that plant carries then the foreign gene and can express it as a new trait. And so this is one way that we can actually genetically modify different crops through gene transfer. With all of this, another aspect of biotechnology and genetic engineering is therapeutic cloning. And really this process is the creation of an embryo to supply embryonic stem cells for medical use. Embryonic stem cells is a topic that we'll look at further and is up to a lot of debate because these stem cells are taken from developing embryos. In humans, removing cells from a developing embryo results in the death of that embryo. And so that has some very big ethical concerns that we'll take a little bit more closer look at. In knowing this and in looking at this further, scientists very recently have developed new ways to develop stem cells for medical use. And why stem cells are important is because they have the ability to differentiate into different types of cells. And so if we start with a stem cell, for example, maybe a patient needs, their heart is failing and they need a new heart. Rather than using a donor organ that may not transplant, that may not be accepted by the body, being able to produce cells that are using stem cells, a heart could be produced for that individual that is an exact match to their body that's not going to be rejected. And the idea behind this of using the genetic information, the nucleus from a patient, combining that with an embryonic stem cell, and then inducing it to become the specific type of cell that you want. That is the process of therapeutic cloning. And so we'll take a look at this and discuss some of these ethical issues in class a little bit more, and also look at induced pluripotent cells that have been produced fairly recently within the last couple of years, and look at some of these different issues in class through discussions. So that's a quick introduction of some of the different uses of genetic engineering, biotechnology, specifically focusing on medication in the medical field, and also for genetically modified organisms, and in DNA profiling, going back to our first video for this unit.